This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 463, recorded on October 13th, 2017. This episode is brought to you by Blue Apron. Blue Apron is the #1 fresh ingredient and recipe delivery service in the country. Get $30 off your first delivery and free shipping by going to blueapron.com/TWiV. This episode is brought to you by the Defense Threat Reduction Agency, part of the U.S. Department of Defense. The agency's Chemical and Biological Technologies Department hosts the 2017 Chemical and Biological Defense Science and Technology Conference to exchange information on the latest and most dynamic developments for countering chemical and biological weapons of mass destruction. Find out more at cbdstconference.com. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me here in New York City, Dickson Despommier.
Good afternoon, Vincent.
Where the weather has taken a turn for the cold, hasn't it?
You know what? I welcome this, actually. It's a little windy out there, and it's in the 60s, and it's overcast, but compared to what we had last week, this is blessings.
18 Celsius.
Yeah, I'll take it.
I don't like it.
I'll take it.
But I have no choice.
No, I'll take this over what we had.
Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. How you doing?
Hey, Rich.
Is it warm there?
Yeah, I'm just looking it up. I'm a little. It says it's 85.
Oof.
I've been standing out in the sun, and that's it's all of that.
Maybe I should move to Austin.
85, partly cloudy. It's actually, I'm seeing mostly sunshine.
And the humidity?
Good.
The humidity is the thing that kills you.
Oh, man.
And that's last week here. It was just awful.
It's great.
It was, I actually, as we left a meeting last night, I commented to my wife that it felt like Florida.
Yeah,
59%, dew point 69. So it's not stinking, but there's some water in the air.
Exactly.
From Western Massachusetts, Alan Dove.
Good to be here. Been a couple of weeks.
Yeah, it has. Missed you. Everything well? Are you
Sound
well?
the same.
Things are okay. And it is 16 C, which, as we know, is 61 Fahrenheit here. It's a lovely early autumn day and pretty clear blue skies. Just really nice.
Got the oval cleaned up and ready to go, Alan?
Got the snow removal equipment. I just got a new pair of winter boots. I got some nice muck boots that come all the way up, almost to my knees.
If you prepare, it will not snow.
I hope.
From southeastern Michigan, Kathy Spindler.
Hi, everybody.
Hello, Kathy.
Temperature here is, well, depending on the app, 66, 67, or 63, which is 17 Celsius. I think I heard from the Farmer's Almanac that it's supposed to be a bad winter, Alan. So I think it's good that you're prepared.
Dickson, you were going to say something?
Yeah, but I forgot it.
Okay. All right, we have a few things to tell you. First of all, Jonathan Udell. Was he on TWiV? I don't think so, was he?
I think you got him queued up for some time.
TWiV, let's see, TWiV, YEWDELL. I know he's coming up in November. Yeah, TWiV 208, the Biomedical Research Crisis with John Udell. He sent me a book he's written.
Nice.
And he wants everyone to have it for free. It's called...
Even nicer.
It's called Truth Wins, a Practical Guide to Succeeding at Biomedical Research. And part one is a recounting of his career from high school, college, medical school, grad school, postdoc, tenure track PI, and tenured PI. That's 7 chapters. Part two, a practical guide to succeeding at biomedical research. Chapters include honoring the scientific method, becoming a life scientist, doing experiments, playing well with others, writing and presenting postdocs, starting your own lab, and finally, what's wrong with biomedical research and how to fix it. Okay. So we will provide 2 links, one for a EPUB version and one for a Mobi version, which you can download for free and read on whatever app you like to read with. Highly recommend it. And he'll be on November 7th, so we can talk a little bit about it.
Nice.
With his son.
Oh, my.
They're both biomedical scientists. Secondly, Gabriel Victora. He was on a TWiV called Concerto in B 161. Man, that's a long time ago.
Long time ago.
That is ancient history.
He has been awarded a MacArthur Prize,
How lovely.
which is, I guess, a good amount of money, right?
It's $200,000 a year
For how long?
for 5 years.
$1 million bucks.
I think so.
It's also called the Genius Award, right?
Yeah. Mm-hmm.
Yep.
Okay, which means you and I, Dickson, will never get one.
That's right. That's exactly right.
Anyway,
Well, I think,
congratulations.
they try to get people early in their careers,
Yeah.
because part of the rationale of the MacArthur Prize was the MacArthur, the founder of it, was a real estate developer who'd made gobs of money, and he's noticed that the Nobel Prizes were usually being given to people who were, about to die and had done their great work 30, 40 years earlier, and thought, well, it would be better to identify these people early and give them money when they need it. And so that's kind of the concept behind it.
$625,000 paid over 5 years.
Oh, okay.
No, it's not $1 million bucks.
That's still pretty good.
It's not bad.
Still pretty good.
And you can use it for whatever you want.
So Gabriel is at Rockefeller.
Yes.
He went to MIT, to the Whitehead, for one of these fellow programs, and
Nice.
he was originally at Rockefeller as a postdoc, and now he's back as a PI. And I actually asked him if he would do an immunology podcast with me, and he said no.
He said no.
He said he didn't think he would have the time. I said, once a month? He said, no, I don't think so.
No.
So, all right, we have some follow-ups from Jess, who writes, hello, TWiV team. As always, appreciate the show, especially episode 461, since so much of my time involves reviews of similar experiments. 461 was the last one we did together with the self-inactivating rabies vector. Thank you for acknowledging the papers are not easy to decipher. When reviewing for safety of the vectors, safety of the transgenes, safety of the procedures, there can be a lot of cross-checking to learn about every single piece of the experimental setup, particularly the molecular biology. During 461, a couple of things came to mind. Apologies if this is a naive question, but maybe other listeners were also curious. Why does doxycycline delay the degradation of a virus? I don't know that you said why, and I just missed it, but the mechanism was lost on me, but piqued my interest. Is it related to ubiquitin? Well, the way it worked is they conjugated a signal to one of the viral proteins, which would direct it to a proteasome for degradation. And they also included a viral protease called the TEV protease, which would cut off this proteasome directing signal, therefore not allowing the virus to be degraded. And that protease was inducible by doxycycline. So you turn on you put in dox, you turn on the protease, you take it away from the proteasome, and that's why it delays the degradation.
Not a naive question at all.
Did I get that right?
It's pretty complicated.
Long time ago. Also, do you know much about studies regarding interaction or synergy between these viral vectors? I just wonder, as we wade further into viral vector research, if anyone has seen anything of interest when using more than one modified vector in sequence, like in the experiment mentioned in 461. I don't know of anything except that people try them and see if they work.
Right.
Let's put adeno associated together with rabies. Don't think about it too much. Just do it. If it works, that's great. If not, move on.
And you'll never find out about the ones that didn't work.
Nope.
Because they won't get published.
Lastly, would it be possible in the future to pull out a list of books that you've recommended, either in picks or in conversation? I often listen while I drive, and if you don't call a book out as a pick, I have trouble finding the books you mention in passing as being good reads once I get to my destination. Okay, so we do put the picks on a page.
Yeah, we do.
But the things we just kind of pop off in the episode, no, those are kind of like extra goodies that we don't list anywhere. I guess I should, but I don't. I'm sorry.
You used to, the books used to be collected in a place on Amazon, right? But that's no
they stopped allowing you to do that. Now all they do is, if you want to put a book on a page, they give you a link, which gives you Amazon affiliate credit if anyone buys it, but there's no way to collect them all on a page. Like in the old day, it was nice because they had a little thumbnail of each book.
Right.
Yeah.
And you could go through and say, what do I want to read today, right? Oh, this is Dickson's pick. I'm not reading that.
Nope. Don't.
Okay, unrelated to scientific papers, regarding gender of letter writers, authors, just wanted to raise the issue that although the comments and banter on the show is based in an effort to respect the letter writers, it is often discussed in the context of a gender binary. A benefit to having several pockets of progressive communities in this current divided political cultural climate is that more people are openly identifying as not belonging to the traditional cultural gender constructs. Many people don't want to identify on one side or the other of the gender binary and reject the notion of gender altogether. Granted, it is common to speak about individuals based on the culturally accepted norms. I just want to raise the issue that maybe the conversation in the future can include the possibility that the person in question doesn't subscribe so as not to exclude someone in that category. Thank you, Jess. Okay.
Sure, why not?
Yeah, that's fine.
Yep.
Which we'll try to be gender neutral. In fact, the next email also discusses that. Kathy, do you want to take the next one, please?
Sure. Jay writes, does it really matter what a writer's gender is? If in doubt, use their name to refer to them or use they, their, them. Simple, uniform, generic, no chance of offending anyone's sensibilities, and emphasizes that it really isn't important in the given context. If it makes someone uncomfortable due to grammatical pedantism, well then, welcome to the future. Jay. P.S. Longtime listener, first-time writer, newly transitioned HIV behavioral research data manager at ISMMS. Thank you all for the incredible program, as well as the other TWiX podcasts. And he has a listener pick. I guess maybe we'll bring that up later.
Sure.
And I agree that it's nice to try and use those alternative pronouns, but yeah, I'm going to be pedantic about this, but it's ambiguous when you're writing about someone and it's a single person and you use they, their, them,
Yes.
and the recipient doesn't know why you're doing that. It can be really confusing.
How many people are we talking about? Yes.
Yeah, and so that I find that a struggle. Plus, this is what I've been doing for many decades now, more than I'd like to say, and it's pretty hard to unlearn, but I'm trying.
Maybe we should just use their initials.
Well, we could say the writer.
Or their initials, just
Yeah.
say JC or...
So often what we do is when we don't know the gender, we usually say it's a guy, and then it's not right.
Mistake.
So then we have a little discussion about what it is, and that's part of this. But I think maybe if we just said the writer when there's any question, right?
Right.
Or the name. Pete, Julie says this instead of he or she, right?
Exactly.
Yep.
Right.
I mean,
it's
the
hard
problem
to do
is
that. We're just so used to using pronouns.
We speak a language with gendered pronouns. Not all languages have that, but ours does.
Yeah.
And so when you try to work around them, everything gets cumbersome. It's cumbersome for these people who are non-gender binary to live. So maybe we ought to kind of suck it up and try and adapt.
I moved my daughter into her dorm last fall. I said, "Where's the bathroom?" She said, "There's a gender-neutral bathroom right there." So I went to it, and it had a big sign on the door, "Women only."
Oh, really?
Yes.
Jeez.
So she was more progressive than the dorm was.
You did the Phil Sharp episode for TWiV 462, which was when you interviewed him for the textbook.
Right.
And he was one of the two people who won the Nobel Prize for splicing. And this is the 40th anniversary of the discovery of splicing. And so there's a meeting coming up at Cold Spring Harbor that it's October 22nd to 25th. Registration deadline is well past. It includes Phil Sharp, Joan Stites, and Mila Pollack, and a number of speakers who then or now have worked on lots of aspects of splicing. So just wanted to point that out. I enjoyed the TWiV, hearing Phil Sharp's interview that you did. And
yeah, I wish I could say that I did it thinking about this anniversary, but it was coincidental. But it's a good coincidence. I didn't know it was the 40th anniversary, although all I had to do was do some math, right?
Right.
I just remember one day in 1970, let's see, when was it in Peter Palazzi's lab? 76, I think.
It was the summer of 77. People came back from the Cold Spring Harbor meeting.
Yeah, 77. So Peter went to that meeting, and he came back, and he said, "This is the next big thing. It's called splicing." I said, What? And he took a piece of paper and drew out, and I didn't know what he was talking about. But I remember that. This is the next big thing. It's nice when, can you say that nowadays? This is the next big thing? What today, Dickson? What's the next big thing?
CRISPR.
No,
that's
I
already
think
here,
an
though, right?
implantable
Well,
chip in your brain that obviates the need for computers and that sort of thing.
Well,
To make a cyborg memory.
We'd have to go to a meeting and see. But
I've heard about this already, by the way,
you've heard of what?
about creating cyborgs out of all of us by implanting memory chips inside of us, which allows us to access anything, anytime, anywhere. I'm not kidding. I'm not kidding. No, it's not a ways off. It's very close.
It has to interface with your brain, though.
Yeah, it does.
Okay,
That's the hard
Kathy,
part.
what were you going to say?
Well,
I was just going to say, I remember when somebody came back from the Cold Spring Harbor meeting, and at UCSD, they gave meeting reviews, and they told you what happened at the meeting. And usually they were quite boring, or you couldn't follow them, or whatever they got excited about really wasn't. But this one was really interesting and exciting. And then the other one I remember, the next big thing kind of moment, was when we did a journal club when I was at University of Georgia. And I think Lois Miller picked and did the paper, but it was about PCR.
It's hard. I'm not.
I remember, I may have described this
here we
before,
go.
but in 1976, when I was in London, Fred Sanger came down and gave a seminar on DNA sequencing.
right.
And that was the biggest jaw dropper I've ever seen.
Well,
It was so elegant. That in the end, you just read the sequence up this gel. Amazing.
I guess when reverse transcriptase was discovered, you could say that's the next big thing because it would lead to the biotechnology revolution.
Yeah, I got
right?
you all beat, though. Because,
What do you got?
Well, because I'm old enough to have preceded the dictum that DNA is the genetic material, so they were still arguing about
Do you remember the structure of DNA in 1953?
Yeah.
When that was announced, how old were you? 20?
No, I was 20 when I was 13.
You're too young.
I was born in 1940.
Too young. Probably didn't make any sense to you.
No.
Yeah.
But it did just shortly after that, because then I got to college and then I had a teacher that could absolutely frost anyone's cake by refusing to believe that RNA was made from DNA. She said, no, that's not right. It's DNA is made from RNA. I said, God.
Yeah, the central dog.
She was teaching us genetics. And
I remember the as the microRNA findings trickled out and then started to coalesce. That was a pretty big deal.
Yeah. I mean,
Yeah, the question is, when do you notice that something is going to be seminal for all fields? Like, splicing.
how about sequencing
Usually it
all of
hits
the
me
life
when I'm
forms
writing.
on Earth?
It usually hits me when I'm writing an article about it.
Yeah. I mean, I'm not.
Because I'll be doing something on a field and I'll say, wait a second, they found what? Wait, what? Wait.
So, I mean, how dare we think that we could sequence every living organism? But guess what?
Were we there yet?
We're heading towards that.
You think?
direction. Yeah.
But we haven't
Well,
even
it's
found-
getting easier and easier because
I
we keep
know.
wiping them out, right?
Well, that's one way to look at it.
Eventually, there'll only be a few dozen species that we need to sequence.
Yeah, but we haven't found them all yet. There are plenty of species.
No, but the
All
point
right,
is
but that gets simpler too, right?
there's-
You just
there's-
eliminate the habitats.
they're so redundant in some ways that the organisms that precede the ones that you want to do, 90% of what's there is already in the next group. And there's 10% difference or whatever that is.
I don't think that's such an impressive thing. I think it's great,
I'm
but
very impressed with that.
I don't think it's so impressive. And in fact, it's not even that accurate, it turns out. I found a paper recently that compared metagenomic sequencing approaches.
I don't want to hear
And
this.
there's so many variables. Almost everything affects the
No, that's probably sure. I mean, sure.
Can we talk about Nobel Prizes?
Sure.
Well,
Yeah,
eventually.
please.
Two of them here in New York City.
That's right. One of them right here.
One of them right here at Columbia. So there are two categories I want to just mention, talk a little bit about. Chemistry, Jacques Dubochet from the University of Lausanne, Joachim Frank right here at our medical college, and Richard Henderson, the MRC Laboratory of Molecular Biology in Cambridge for cryo-electron microscopy.
Yeah.
Now, Joachim Frank, I have never spoken a word to.
No,
We have three Nobel laureates here, right?
do.
Frank, Richard Axel, who I have spoken to,
Yes, and Eric
Kendall, who I've never spoken a word.
Right.
Oh.
Never. My fault.
I've even spoken to his wife,
Who?
Eric Kendall's wife.
Dare you? How dare you do that?
No, she's one of the professors.
Anyway, So Joaquin Frank is like almost 80, right?
True.
And everyone tells me he's a lovely man, but I've never spoken to him. But he was recruited here from Albany a number of years ago. And so there's an article on the nobelprize.org. Lovely. If they give you a bunch of articles, one is a basic one and one's called advanced for each prize. So I have the advanced one. It's a great article that traces electron microscopy from its very beginnings through cryo-electron microscopy. And they talk about what each of these individuals did.
Right.
And they have a nice summary here, which I'll just read briefly. Single particle cryo-electron microscopy is unique, does not require crystallization, uses very small amounts of material, covers a wide range of sizes from particles the size of hemoglobin, which is 64 kilodaltons, to very large particles like viruses. Cryo-electron tomography is used to determine structures of even larger objects, including organelles and cells. So you got a big range of things you can study. Now, this article is great. It starts with X-ray crystallography, how they used to use negative staining to see things and the artifacts associated with it. And they couldn't really determine any high-resolution structures, so they went to X-ray crystallography to do high-resolution structures. But then people started developing ways to visualize without negative staining. First high-resolution cryoEM structure was in 1990, Bacteriorhodopsin. Okay. Now, what Frank did, one of the things he did, a fundamental problem in studying unstained, non-crystalline, asymmetrical, randomly oriented particles is, quote, the alignment of features that are only faintly visible on a noisy background. So in the 70s, Frank addressed this problem by presenting a method for aligning low-dose images of individual molecules using cross-correlation functions. So that's part of his contribution to the.
So I heard a really nice seminar on cryoEM just the other day that put this into perspective. So if you take a picture of a three-dimensional structure, the image that you get out is two-dimensional, but it's got all of the three-dimensional image in it. It's just the three-dimensional information. It's just squashed. And Frank figured out two things. First of all, how to take a bunch of, and you'll get them in all different orientations.
Right.
Okay. Frank figured out, and the computers that do this, how to take all the things that looked in a similar orientation and average them, which increases the resolution, and then take all the different orientations and compare them. And that allows you to construct a 3D image out of a bunch of 2D images. Similar in a way to tomography, I suppose, except you're not actually tilting the stage. You're just using the natural multiple orientations of things to get the 3D structure.
I didn't realize he had done this. Did you, Dickson?
Didn't even know he
so
was on our faculty.
They say in this little description, he made these tools, as Rich said, these computational tools for image analysis. And he gathered them together in a suite of computer programs called Spider, making them readily available and usable for the scientific community.
Right.
So that's very nice. Henderson developed a new preparation method that enabled studies of unstained crystals in the electron microscope at room temperature. He replaced water with glucose to preserve samples in vacuum at low temperatures so that they could do the freezing. And Dubochet presented a method that allowed formation of a film of non-crystalline solid water on a specimen grid for observation, so you could take these pictures. So
that's where the cryo comes from. He's responsible for the they call it vitrification. And what I didn't realize is, I realized that, so the whole thing is you got a sample suspended in water and you stick it on an EM grid, and then you dunk it into something really cold. It's like ethane that's chilled with liquid nitrogen. So it's on the order of minus 196 centigrade. And the instantaneous freezing allows it to freeze without forming ice crystals, which damage the sample and mess up the image. What I didn't realize is that freezing is not really the right word. It's like a super cooled liquid.
That's why they call it vitrification.
Right? Yeah, vitrification.
Right.
Yeah.
So it's still it doesn't move anymore because it's so cold, but it's still water, still liquid.
Yeah, because initially they just froze it and it damaged things. It didn't work, right? So they had to pick up that. So,
Right.
I was up at Cornell a while ago, and someone showed me that they have a special thing for doing this vitrification, and it's an expensive. It's like $50,000 for this little thing that's on the bench, and you dip in your samples, and then you put them in a container, and you walk them over to your microscope. Man, everything's expensive.
And then, of course, you got to have a microscope that stays cold.
Yep.
Otherwise, everything evaporates under a vacuum.
Right.
Correct.
Right.
So I understand that when Frank came here, these microscopes, he got the most powerful one you could buy. I don't know how they measure the power of a cryoEM scope, but
That's electron volts. It's
Electron
measured by
volts.
Electron volts.
Yeah.
and maybe a megavolt. Is that possible?
Oh, yeah.
Yeah.
And they bought it for him, and now they got a Nobel Prize. I saw a picture of him with President Bollinger and Dean Goldman, right? All in the same. It's the only time they would get together with an investigator if they get a Nobel Prize.
That's right.
It's kind of sad.
Well, yes.
Anyway, congratulations. The other Nobels that are interesting to us, well, at least to me, maybe others like the physics and the others, but that's not what we talk about on TWiV. Physiology or medicine. Jeffrey Hall from the University of Maine, Michael Rosbash from Brandeis, and Michael Young from Rockefeller for circadian rhythm genes. Mm-hmm. This is amazing because I just read Time, Love, Memory. We just interviewed the author Jonathan Weiner for TWiVo, and he tells the story of these three individuals in this book, which was written some time ago. It was written in 1999.
Precedes
Hall, Rosbash, and Young.
I'll be darned.
And I emailed him. I said, "You must be happy." And he goes, "Oh my gosh, when I heard this, I felt like I had just drunk 6 espressos." And I just want to tell you, there's some cool things in this book. So this whole story starts with Seymour Benzer. Did you know Seymour, Dickson?
I knew of him.
I never met him.
I was here while he was here.
He started out, he was a phage geneticist.
Yes.
It was famous for the phage R2 protein mapping like every base by mutation. He's a geneticist. And then he moved to Caltech, and he wanted to study the nervous system. And he looked for a system to study, and he ended up with Drosophila.
Yep.
Now, in this book, Weiner describes how the work of Mendel was forgotten by the early 1900s, and Thomas Hunt Morgan here at Columbia revived it by trying to use Drosophila as a genetic system. He actually identified the first mutant of Drosophila called White, which had white eyes. He didn't use any mutagenesis. He just kept screening offspring until he found a mutant. Later on, they figured out how to zap the flies. Mm-hmm. So Benzer said, "I want to study neurological problems." And the title of this book is Time, Love, Memory. Those are the 3 things he talks about: the clock, what genes are involved in sexual attraction, what genes are involved in memory. And people thought he was crazy. They said, "There's no one gene that's going to affect any of these things." But he said, "Yeah, I think so. I'm going to try it." And so he had this postdoc called Konopka. Let me see what his first name was. What is his first name? I can't remember his first name. Frank Konopka. Here we go. No, it's Frank Brown. Sorry. All right. I don't know what his name is, but he was a postdoc, Ronald Konopka from Dayton, Ohio. And they said, let's find flies that have an altered clock. And everybody in the lab said, it's not going to work. You're never going to get this. And Konopka said, the hell with you. He said, bugger off. That's what it says in the book here.
Or bug off.
He said, bugger off. That's what it says here in the book. They said, if you make one, it will die. But he told them, bugger off. So what he did, he kept them at cycles of light and dark, and the flies would eat close during daylight. And he just looked for flies that would eat close at the wrong time. So in the morning when he came in, if some flies had laid eggs, he'd say something wrong with them.
Bad timing.
So he went through, he went through many flies. He was mutagenizing them. In bottle after bottle, the flies behaved normally. In the 200th bottle, he saw it was teeming with flies. And when he inspected them one by one, he said most of them were males. He bred them. They also had a screwed up clock. So he identified this first clock mutant. Mm-hmm. And he then Benzer started giving a seminar on this, and Max Delbruck came up to him at one point and said, "I don't believe a word of it." He was typically skeptical. All right, so now they had this first clock mutant. Hall was a postdoc in Benzer's lab, and Hall was studying sexual attraction between flies genetically as well, and identified some interesting genes. And he went to Brandeis to be a faculty member. And there became friendly with Rosbash. And so Hall was a Drosophila geneticist, and Rosbash was basically a molecular biologist who, and at this point, all the cloning methods were being developed, and the two of them got really friendly. Apparently, they liked sports, and they played basketball together and all that. And that they got together and said, "We should clone this gene." And Hall was like, "This is going to be the most important gene in history. We have to clone it, this thing that controls the circadian rhythm." And so they had mapped it next to the period, next to the white gene, which happened to be X-linked. And so what they did is they chopped, you could see the chromosomes in flies. They basically cut out pieces visually and cloned them and did an expression experiment and cloned the gene that way.
Drosophila was chosen.
And then they got the gene, they called it period, and the protein was called PER. Now, at the same time, Michael Young was at Rockefeller doing the same thing. And they were racing each other. Hall and Rosbash got their paper accepted for the issue of Cell December 84. At the same time, Young and his group got their paper accepted for the year-end issue of Nature that straddled December 84 and January 85. Afterwards, they said to him, "Well, yours is 85, really."
So
Ours
even
is
then,
84.
timing was important.
Of course. So that's why the three got the prize. And of course, after that, they studied the protein and figured out why it cycles and all of that. Lots of interesting studies from all of the labs. But I highly recommend now you read this Time, Love, Memory, because it's a great accounting. He interviewed all these people and has nice stories in there and so forth. So I would check it out.
And as is always the case with the Nobel Prizes, all the other people who were involved in this work receive bupkis.
I thank you for mentioning that. Konopka, who identified the first...
Yeah.
He never got tenured at Caltech.
Ooh.
He ended up at Clarkson.
Yep.
And I don't know if he's still there, but he's not even,
There's a lot of stories like that, unfortunately.
I mean, without him, they wouldn't have been able to do this.
Sure.
Right? But you can only give it to three.
It's true.
Back when you mentioned that Seymour Benzer left the phage work and went to Caltech, he left from Purdue University, and he was working in Lilly Hall of Life Sciences. And when I did my undergraduate research, there was a shelf in the cold room that was labeled Benzer.
You'd already left, huh?
Yeah.
I got to go, gang.
Okay, Rich.
See ya.
Bye.
Have fun. See you next time.
Yeah, nice talking to you.
Bye.
Later.
Bye. Alan, can you read? So at the same time, we got an email about this from Neeraj. Can you read that, Alan?
Yes. I just as Rich was leaving, I had to evict a cat from my desk. Pardon me.
Oh, leave the cat there. No problem.
She decided she was going to get in front of the screen and step on the keyboard. It was getting to be a bad scene. Okay. Neeraj writes, "Hi, Twivers. This is Neeraj from Sutravax. I've been meaning to write for a while, but it hasn't happened of late, so here it is. I hope all of you are doing great, and hopefully it's been yet another year of memorable scientific experiences. First and foremost, many heartfelt congratulations for completing 9 years engaging in this incredibly generous undertaking. I'm personally most thankful to Professors Racaniello and Despommier for having done both TWiV and the TWiP series. These have just been incredibly information-dense and fact-rich podcasts for me, which I can listen to over and over again. Not to say at least how much I have enjoyed the amazing chemistry you both share. A major investment like these can't only be a product of curiosity and incentive, but of personal admiration, too."
Wait a minute. What's this chemistry?
Acid and bases.
Yeah.
You've made a complete acid out of yourself.
He didn't say it was good chemistry, though.
He didn't say that. That's true. He didn't.
Redux chemistry. All right.
No, come on.
Today is even more special for another reason. My alma mater, Rockefeller University, posted another major achievement to their crown jewel. Professor Young was awarded the Nobel Prize for the discovery of circadian rhythms, which is something I've always been interested and fascinated by. I was lucky enough to have attended a few of his seminars during my grad school days. This is one of the primary examples where there is a direct and clean correspondence between protein physiology. And not to say that this field also has some of the most appropriate and fun names for the proteins involved, namely timeless, double time, and period, hence the reference in the title. In relation to this, I was also happy when you recently brought in Jonathan Weiner, who's the amazing writer of Time, Love, Memory, one of my all-time favorite scientific reads. Having spent an amazing 5 years as a graduate student at RU, I'm never surprised by the heights that its faculty scales. With such rich history from generation of the yellow fever vaccine that was discussed a few TWiVs ago to circadian rhythms to discovery of dendritic cells, it's a phenomenal melting pot of groundbreaking scientific discovery. Mm-hmm. My class of 22 graduates had 17 nationalities within it, which bolstered a rich sense of cultural exchange in me and something I've enjoyed ever since. There's so much to learn from each other at the scientific and human level, and RU facilitates that in every possible way. The richness of scientific expertise in the entire neighborhood encompassing Weill Cornell and Sloan Kettering research institutes is just remarkably unique. I personally feel that science alone without able mentorship at times can leave you disgruntled and frustrated, and for this particular reason, I would personally like to commend and thank the entire TWiV crew. Each one of you in engaging in an act of selflessness, which you didn't have to, but I'm glad that you did. I'm absolutely certain that millions like me have greatly benefited from your incessant desire to learn and explore. Thus, kindly keep up the great shows, and here's to many, many, many years worth of TWiVing, TWiPing, and TWiMing ahead, since there's an ocean of information out there, and without a good teacher, students would feel inundated, lost, and overburdened. And like we used to sing during RU's grad student outings, we are, rock you.
We are, rock you.
You guys are the true rock stars. Best, Niraj. Well, thank you very much, Niraj.
And he actually said, here's to many, many, many to the N years.
Yes, right.
Oh, yeah, I didn't see. I thought that was a typo.
Yeah.
Well, I'm not sure millions, but cumulative millions.
No, I would echo all of what he said. Even being a postdoc there for a shorter period, 3 years, was an astounding experience and one that I'll never forget. And just to connect back to what we were talking about a little bit before, I had the pleasure of, as you know, I've mentioned this several times, and I'll say it again now, had the pleasure of sitting and listening to Theodosius Dobzhansky talk about how the original Mendel papers were discovered and how they were translated and why they didn't all match up with the exact data that was actually put forth, because some of it looked to be altered a little bit just to make things come out more even. But nonetheless, the essence of the way these tall and short sweet peas would grow were absolutely astounding. And to see a man who had a direct connection to those papers is like reading someone who just translated the Dead Sea Scrolls. It was quite amazing to just sit there and listen to this history just flow out of him. And he was a wonderful person too. And so he was very sharing with his knowledge. So there.
Rockefeller has had 25 Nobel Prizes.
Yeah. When I was there were 12 people that had Nobel Prizes.
Well, they're all there at the same time.
Yeah, they're all
12 people who don't talk to each other.
Some didn't. They were not all alike, I must say that. But
well, they probably know how to handle the press conferences there at Rockefeller.
Oh, they sure do.
Yeah.
They sure do.
Dickson, can you take that last one from Pete?
Sure. Pete writes, this excerpt from NPR News caught my eye. With the award, 7 Americans have now been honored among the 9 researchers who have been recognized so far in the 2017 Nobel season. Toward the end of Wednesday's event, a journalist asked Royal Swedish Academy of Sciences Secretary General Goran K. Hansen about the prevalence of Americans. In his reply, Hansen noted that since World War II, the U.S. has, quotes, allowed scientists to perform fundamental research, unquotes, and focus on large-scale projects that might not have immediate impact or applications. He added that they have also been able to do so largely without political pressure.
I don't know what he's talking about.
Aloha.
That's just totally wrong.
Pete.
Maybe during World War II and after, but not today. Does anyone think that this is not correct today?
Oh, I still think it's correct to some extent. Not as much as we might like, but compared to
I think
maybe other countries
compared to most other countries historically, since World War II and at least up until very, very, very recently, there has been an immense amount of support for fundamental research in the U.S. And even now, there's still a lot, though not as much as would seem to be required by the population of investigators who want that funding.
Right.
So yeah, we have this whole concept of modern fundamental science was generated to win World War II. A guy named Vannevar Bush prevailed upon FDR to set up the system of grants that would be given to researchers who would stay at their own universities rather than coming to work in some government office. And that was a key part of the arrangement because he understood as an academic himself that professors would want to stay at their own institutions. And he produced this entire grant funding mechanism that persists to this day. And the notion of peer-reviewed grant funding that would be given to the institutions and how that's all structured, that all came out of that one thing. And that has been copied by other countries around the world, but we invented it. And it has been incredibly successful, and it is why the United States became the sole world superpower in the 20th century. Now, we're currently in the process of squandering that at an alarming rate here in the 21st century, but that does not deny that the history of this, and in fact, if you go back to when the discoveries for the Nobel Prizes were made, since they typically have at least a few decades lag, I think that certainly applies.
Yeah, I agree with the history, but having sat through 35 years of watching how you could write an NIH grant about the science and then having to write the human health relatedness of the work and hearing study sections go, what's the human health relatedness of this? It's ridiculous. Now, it may be better still than in other countries, but we shouldn't care about what other countries. We should worry about doing fundamental research here. And I don't think the atmosphere condones that anymore in this country. It may be going on, but it's not as good as it should be.
Well, I'd like to.
We are in the process of squandering the system. We are in the process of taking it apart.
But Vincent, you will agree, and everybody else too, that we have two granting agencies, major granting agencies. Mm-hmm. One of them is NIH, and the other one is NSF.
Yeah, so NSF, small compared
no, that's what I'm going to say. NSF has also program-directed research.
Totally,
right?
I agree. They don't want medically related research.
They do not.
I agree. But there's so little money there, right?
No kidding.
They should be given more money.
I would agree.
Let's give them $30 billion.
Done. What else do you want?
Okay, Don.
A not well-known fact is that in the materials that reviewers get when they're on NIH study section is a document that emphasizes the fact that not everything has to have human translational importance. And our study section, at least the last couple times I was there, made a point of emphasizing that at the beginning of study section. I'm not sure that it makes a difference, but at least there is a document out there that says that.
Well,
and that to some extent, we reviewers collectively have brought it on ourselves.
Yes.
Because we think that there has to be this translational relevance for human health, and that's not necessarily the position of the NIH.
And I think it's a byproduct. This mode of it has to be translational, it has to be critical for human health. I think there is an element of that that's due to the continuously constricting funding, because in real dollars, especially if you do what's called sector inflation for what actually the costs of biomedical research are, the NIH budget has been going down for quite some time.
Sure.
And as a result, there's less and less money, and you're seeing these smaller and smaller pay lines. And I think part of what's happening is that the study sections, I'm not sitting on study sections, but I think there's probably an idea that, well, if we can only fund a tiny number of these, maybe we ought to fund the ones that are medically important. And I think that may be a factor in some of it.
Yeah, I think it has also become politicized, right?
Yes.
I do remember a time when Congress pretty much stayed out of NIH funding. And I remember distinctly during the first Bush presidency when all of a sudden Congress wanted to know where this NIH money was going. And then you had to tell them you're working on diseases like diabetes and cancer and so forth. And it moved very slowly away from basic research. And you got over the years all these criticisms. What are we working on worms? What are we working on flies for? And so the politicization is an issue. And that's why him saying that without political pressure, that may have been for many years, but not recently.
Yeah.
What's really incredible also to realize is that so many of these basic research findings have had impacts on medicine.
Huge.
And, like CRISPR-Cas9, I mean, that's a bacterial thing. Who cares about that
all
stuff?
The
and all of a sudden,
virus replication. That's why we have virus vectors for gene therapy.
Tell me about it.
Without the basic stuff, you wouldn't have any of that.
That's why we even know what causes a lot of diseases.
But at least you say, welcome to This Week in Virology. Viruses, the kind that make you sick. When you say the kind that make you sick, that immediately throws you off into the medical direction. Well, and that as opposed to the paper we're about to do today.
Yeah.
So maybe we're not necessarily all about the kinds that make you sick.
No, we are not. That was just
no, I understand.
Just a tagline.
Tag to make
I understand.
Realize this wasn't about computer viruses, right?
or the ones that make you well.
We're all very much aware that most viruses do not make you sick.
That's right.
Right? Or make anything sick.
Correct.
In the sense of the word. Although a lot of them kill bacteria, so they're pretty sick. But
this is true.
We do. This episode is brought to you by Blue Apron. Blue Apron's mission is to make incredible home cooking accessible to everyone while supporting a more sustainable food system, setting the highest standards of ingredients, and building a community of home chefs. For less than $10 per person per meal, Blue Apron delivers seasonal recipes along with pre-portioned ingredients so you can make delicious home-cooked meals in 40 minutes or less. You can customize your recipes every week based on your dietary preferences, and there's no weekly commitment, so you get deliveries when you want them. They have a freshness guarantee, which promises every ingredient in your delivery arrives ready to cook, or they'll make it right by shipping the exact amount of each ingredient for each recipe. Blue Apron is reducing food waste. This October, Blue Apron is celebrating its 5th anniversary by bringing back its top 20 recipes as picked by the Blue Apron community. And this is for a limited time only, so check it out. Blue Apron is all about giving people fresh recipes to explore as they learn to cook new dish after new dish, which is why they don't normally repeat recipes within one calendar year. That's what makes this limited time offer so exciting. Now, I've tried Blue Apron, of course. I'm not a fan of shopping. I don't like going to supermarkets. I always forget something. So this is great. Blue Apron, the box comes right to the door. It's everything you need. It's all measured out for you. Easy to do. There's a lovely recipe card. You can get almost anyone interested in cooking. You say, hey, let's do this meal in 30 to 40 minutes. Here's the directions. Let's do this. Really easy to do, and it'll be delicious. And the meals that we have made are great. They're just wonderful. I absolutely love it. And for people that are busy or just want to learn how to cook, or even people who know how to cook really well, it's great to try different things. So check it out. Try Blue Apron's all-time consumer favorites. Go to blueapron.com/TWiV to get $30 off your first delivery and free shipping. That's blueapron.com/TWiV. You'll love how good it feels and tastes to create incredible home-cooked meals with Blue Apron, so don't wait. Blue Apron's a better way to cook. This episode is brought to you by the Defense Threat Reduction Agency. Imagine an everyday, inexpensive drone you could buy online, modified by terrorists to spread chemical or biological weapons over a crowded football stadium or a holiday parade. Plague, VX, sarin, weaponized flu. How could we prevent a scenario like this from happening? How would we treat the victims? What could we do to counter the effects? Well, for answers to some of these questions, join us in Long Beach, California, November 28th through 30th for the 2017 Chemical and Biological Defense Science and Technology Conference to exchange information on the latest and most dynamic developments for countering chemical and biological weapons of mass destruction. Collaborate with more than 1,500 scientists, subject matter experts, military service members, industry partners, and academic leaders from across the globe who are committed to making the world safer by confronting chem biodefense challenges. Part of the U.S. Department of Defense and charged with safeguarding our warfighters and our nation, the Defense Threat Reduction Agency's Chemical and Biological Technologies Department is the host of this important conference and brings together the best and brightest from around the world. Please join us to share your important ideas. For more information and to register for this conference, visit cbdstconference.com, and you can stay up to date with the latest conference information over at Facebook. Just look for or search for DODDTRA, or on Twitter, the handle is CBDST Conference. The 2017 CBDST Conference, today's innovation, tomorrow's warfighter capabilities. Here's our paper for today. It's a PNAS paper just published October 3rd. Arabidopsis M6A demethylase activity modulates viral infection of a plant virus and the M6A abundance in its genomic RNAs.
And if you're put off by the M6A in the title and you're saying, gosh, that's cryptic, just you wait.
Right.
I bet this would get a low readability.
This would get an extreme, the abstract would get a very low readability, I think.
It was so low with me, I didn't read it, but I'm about to follow
Anyway,
it now.
this is from the Instituto de Biologia Molecular y Celular de Plantas in the University Politécnica de Valencia, which is, of course, in Valencia. And the first author, well, the authors are Mireia Martinez Perez, Frederica Aparicio, Maria Pilar Lopez Greza, Jose Maria Beles, Jesus Sanchez Navarro, and Vicente Palas. Hey, Vicente, that must be Vincent, right?
I think so.
And I don't know, I thought we would do a plant paper now and then, so let's do another one. We haven't actually talked about N6 methylation of adenosine at all. This happens in eukaryotic RNAs, mammals, insects, plants, yeast, animal viruses. It's been known for a while, but lately it's become hot. In fact, Stacy Warner, one of the winners of the Ann Palmenberg Award at this past ASV, she works on this. She gave a very nice talk about it. So it's methylation of an adenosine residue. It's an N6, putting a methyl group on the sixth, the nitrogen attached to the sixth carbon of adenine. And this modification is carried out by enzymes in the cell, and there are enzymes that also take it off the RNA. It's thought to occur in the cytoplasm in eukaryotes. It is thought to regulate, in terms of cellular RNAs, their stability, translation, and nuclear export splicing, etc. Many different things. It's been not too long ago, people are starting to understand that this modification happens on viral RNAs as well, and it regulates the replication efficiency. Let's put it that way. It's been shown to be, for example, one of the first papers showing HIV replication is affected by this modification, hepatitis C virus, Zika virus. So you put on methyl, but it's not the same for all viruses. For HIV, methylation has one effect, and for Hep C and Zika, it has another effect, the opposite effect. But in either case, it's regulating the efficiency of viral replication. But
this is part of a general theme of methylating nucleic acids as a regulatory mechanism. Methyl groups are just a handy chemical tag that you can stick onto nucleic acids and mark them in some way, and it's reversible. So this is used for lots and lots of gene regulation stuff in the cell.
When you use the word regulation, now this is a point of interest here. You're saying that things were chaotic, then you put this on and now they're regular. That's the way I thought about this.
That's funny. I like that.
Because, there's some order in all of this now, and it's going to proceed at a certain rate. Some rates are higher, some rates are lower. Right. Is that correct?
Well, I don't know that there was disorder before, but there certainly would be without this.
Right. So not adding the methyl group lets things go completely, what?
Well,
stochastic?
Depends on the situation. It depends
sometimes having the methyl group on there makes, as we'll see, can make something more able to be transcribed or translated or replicated or what have you. And sometimes the methyl group will inhibit. So it's context dependent.
So, for example, Dickson, for HIV, if you silence the methylase that adds the modification, you decrease HIV replication.
You do.
If you silence the demethylase, you increase viral replication. But the opposite occurs with Hep C and Zika. So essentially, the modification can have a variety of effects. It can increase or decrease depending on the virus.
Does it depend on the kind of virus or just the individual virus? Like, for instance, HIV is a retrovirus, but the other viruses are not. They're flaviviruses.
I don't think enough have been looked at to make general conclusions like that.
Right. The one that we're going to hear about here is a positive sense RNA virus like HCV and what was the other one? Zika, sorry. So in that sense, with a very small n, Dickson, your conjecture is correct, but we don't know that that's...
And remember, Dickson, this is a modification that occurs on most eukaryotic RNAs, right? Of all sorts, not just messenger RNA, but all the others.
I'm thinking as a person who hasn't read this paper.
And it can regulate their activities as well.
Right, no, got it.
Like stability can be regulated.
Well, yeah, sure.
Right. Now, this hasn't been studied much in plants. That's the thing, that's the interest in this paper. Mm-hmm. And of course, the cool thing is they use viruses to study this. And of course, viruses show the way. Right, we know that.
Always.
And can be used to study cellular processes.
Nature's monkey wrenches.
And they use Arabidopsis thaliana, which I know, Dickson, you're familiar with,
I
right?
am.
Have you eaten it before?
It's not an edible plant. But no, it's a very adaptable flowering plant. It's an angiosperm.
And it's commonly used in laboratories, right?
It was called the Drosophila of the plant world.
Mm-hmm.
That's exactly right.
So we know that the mRNAs of Arabidopsis are, in fact, do have this M6A modification, over 2/3 of the RNA. A methylase homologue has been identified in the genome. Demethylases have also been, no demethylases, just methylases. No demethylases in this plant have been described.
Okay.
But there are homologues of an E. coli demethylase, but they haven't been functionally characterized. So here they study viruses, 2 viruses. Alfalfa mosaic virus is one of them, and that, of course, sounds to me like a virus of alfalfa. Mm-hmm. And cucumber mosaic virus. And both of these viruses can infect Arabidopsis, although that's not their natural host, I would guess, given the names, right?
Right.
Now, the first thing they do is they take the capsid protein of alfalfa mosaic virus and they identify an interacting protein, which turns out to be a demethylase. So they take the capsid protein of the virus, they use yeast-2 hybrid approach to say what Arabidopsis proteins interact with it. And they pull out this demethylase, which has the wonderful name ATALK BH9B.
Just rolls off your tongue.
Yes.
Happens to be the Yes, Kathy.
Say that 3 times and it's yours.
Just call it 9B.
Hey, 9B would be good.
They should
9B
come
would
up
be
with a clever name.
As I read this paper, I thought, so what are you guys going to call it? No, what are you going to call it?
Right.
And throughout the paper, it's ATALKBH9B.
Yeah, always copy paste.
Yeah,
copy paste, right?
It's quite a. I think 9B. I was saying AdAlk. That would be another one, but 9B. We'll call it 9B, and we'll call it 9B.
Because there's also other things. There's 1 through 10B.
Yeah,
right.
Anyway, so this is a candidate by homology with E. coli.
It's a candidate password, is what it is.
It's a candidate password. That's a good
right.
password. I wonder if it has a high entropy. There are not a lot of non-letter characters in there, though. All right, so they confirm that this, in fact, does the protein encoded by this gene does interact with the capsid protein by a variety of approaches, and then they identify a strain of Arabidopsis. You can get these that have a transposon insertion in various genes. They find one where the 9B gene is knocked out. I remembered 9G. So you can just go to this Arabidopsis stock center,
Right.
and look through the catalog and say, oh, here's one with a disruption of the gene we're interested in. They get those plants and they infect them with alfalfa mosaic virus particles. And they find that the absence of this 9B gene, the absence of the 9B gene, what does it do? You have fewer viral RNAs compared with wild-type plants.
So it downregulates viral
So
replication.
normally if you have the gene there, if you have 9B there, it positively regulates infection. And this is a candidate demethylase.
Right.
So already we could say maybe taking off methyls is good for virus replication. Makes sense?
Keep viruses off meth. Yep.
To be clear about the mutation, it's not that the gene isn't there, it's that it has this insertion in it. So
Right.
it's a mutant.
It's disrupted.
Wild type versus mutant. And we're not going to say that the mutant is bad. It's just
It's just different.
Is anybody confused over the fact that you can take Arabidopsis, which is not alfalfa, and infect it with the alfalfa
No,
mosaic virus?
I'm not confused.
No, I mean,
The virus has a broad host range. Why
well,
not?
apparently.
All right. So what kind of plant is alfalfa? Let's see if we can.
Grass.
And what kind of plant is
It's a
Totally different?
Yeah, it's an angiosperm, but it's a totally different family of plants than alfalfa.
Well, I tell you.
Totally.
Broad host range.
Because
alfalfa.
some viruses are very, very narrowly focused in plants, like wheat, like rice blast, for instance, et cetera, et cetera. I mean, some are very host specific, and some apparently.
I think
are
it's
not.
an interesting
Is
question.
alfalfa a grass?
Well,
alfalfa's in the pea family.
it is.
Yeah, I know it's often part of hay, but it's not actually grass. It's not a C4
All right.
grass.
So this virus can
I said
infect
corrected.
and cause disease in a wide variety of plant species.
Okay. That's
Okay, so
right.
has a wide host range. Alfalfa probably reflects the plant where it was first discovered, I would guess, and
Right.
it
I guess.
can do a lot more than alfalfa, right?
Right.
So
It can infect all the little rascals.
so we have
Exactly.
a plant.
Buckwheat.
Did they look at this plant with the 9B transposon? They have reduced levels of 9B mRNA expression. So it's probably not zero. It's just reduced. And in those plants, you have less viral replication. This protein encoded by the 9B gene, they did some localization studies. They find that the protein's in the cytoplasm, and it locates to small cytoplasmic granules. And these appear to be what are called P or processing bodies, which is kind of a purgatory for RNA. You're sent there.
They're purgatory bodies. That's the key.
You're sent there to be silenced,
to do penance.
to be processed, to be degraded. So it's kind of a turnover place. So the suggestion is maybe this demethylase is involved in modulating levels of RNA. But I think the key experiment here is that they actually ask, is the protein a demethylase? So far we don't know. So they have an assay for this. They make the protein and purify it, and then they mix it with single-stranded RNA that has been methylated, M6A modification. And then they treat it with the enzyme, and they find that the enzyme completely demethylates the RNA. So M6A, that specific M6 adenosine modification, is taken off by this enzyme. So that's very nice.
So it's no longer a candidate demethylase.
It's no longer a candidate. But listen to what they say, and I knew this was going to sound pedantic, but I know Kathy would like to hear this. They conclude, therefore, 9B is a protein described in plants with single-stranded RNA M6A demethylase activity. Why isn't it a plant protein? Why is it described in plants, right? I know I'm being pedantic, but it sounds weird.
Yeah. Yeah, I don't know. They're maybe not. It's because they're not native English writers.
Yeah, it's okay.
And I think they're harping on the fact that people have not looked at this methylation phenomenon in plants.
Right.
They knew that they had these genes with homology and that there were
Right.
10 of them or so, but they had just never shown the functional.
Yeah.
So they're just reminding us yet again, hey, look, guys, we found this in plants.
So the other question, of course, is alfalfa mosaic virus RNA methylated and M6A methylated, which we haven't looked at yet. So they look at that. They extract RNA, viral RNA, and they have an antibody to this methyl, this M6A methyl, which is very nice. It's specific for that. So they can immunoprecipitate the RNA, and they can, I think they digest it and sequence it even and quantify it.
They look at the amount of M6. This assay I did think was kind of cool. They first did it with their GST tagged 9B protein. They incubate it with an oligonucleotide to which they've added M6A, and then they treat it with benzonase, phosphodiesterase, alkaline phosphatase, and then they do mass spec on it. And they do the same thing with the RNAs from the infected plants and then the RNAs from the virions.
Yeah, they use mass spec. In the old days, we would have used paper chromatography. I don't know if you remember those days, Kathy, but
oh yeah,
we would have had very smelly solvents in big glass tanks with papers or solid matrices in them. We would scrape off the dot, count it. Oh my gosh. Yeah, mass spec is much easier. Of course, it's more expensive. So they do show that the RNA is methylated. I'm trying to see what the hell else they did here. Did they digest the RNA with the demethylase? I don't think they did that experiment, right? Oh, they sequenced. They immunoprecipitated with their M6A antibody, and then they sequenced the pieces that were brought down to see the sequence of the genome where the methylation was occurring. And they find six peaks distributed along the genome where there's amethylation of this sort. They look at cucumber mosaic virus. They find that this viral RNA is also M6A methylated, but the 9B disrupted plants, the mutant plants, if you will, they can still be infected by this virus as opposed to alfalfa mosaic virus. So this virus, cucumber mosaic virus, doesn't seem to be affected. Its genome is methylated, but doesn't seem to be affected by the absence of 9B protein. So obviously there are other methylase-like genes in the genome, so maybe they're involved as well. So I think I said at the beginning that this 9B protein interacts with the alfalfa mosaic virus capsid. The cucumber mosaic virus capsid does not interact with it. However, they have another virus they look at called Prunus necrotic ring spot virus, closely related to alfalfa mosaic virus, but it doesn't replicate in Arabidopsis. There you go, Dickson. There's a plant virus that doesn't. Prunus.
So a lot of them have specificities.
Yeah, some do, some don't.
Yeah, that's right.
So they could, the capsid protein of Prunus does interact with 9B protein, but they couldn't assay the effect of it because they couldn't.
I'm surprised Prunus isn't more lax.
Oof. Oof. Very good.
This is 6XSSC. The diseases that they cause, though, okay, they're called mosaic diseases.
Yeah.
They're called that because patches of depigmentation occur throughout the leaf structure of these plants. So does that suggest that there's a patchy distribution of these enzymes that demethylase or methylate?
Not necessarily. No, that could be for some other reason, right?
I don't know.
What is a necrotic ring spot, Dickson?
No idea.
It doesn't sound like a mosaic.
It's not a mosaic.
And it's necrotic.
But the mosaic, I'm talking about the mosaic.
It's a different pattern. It's a different pattern of pathology.
Yeah, exactly right.
I mean, but I think the
no, lots. They're very descriptive in their terminologies for these plant virus diseases, though. Banana wilt, right? Wheat rust.
Yeah, it's great. Love it.
Yeah, I like that a lot. I actually
compare it to poliovirus.
Well, you wouldn't guess what it did.
Well, poliomyelitis, right?
Right. No. Myelitis. That's
true. Do you remember the first virus discovered ever?
Tobacco mosaic.
Yeah, it's another mosaic, right?
That's right.
So here we have the thing here, the summary here, if you will. They have found 2 plant viral RNAs whose RNAs are modified with this M6A. They found a demethylase encoded in the plant genome, which regulates viral replication. So if you take off the methyl groups, the virus does better. If you take away the demethylase, the virus doesn't do well. Huh. So when the virus genome is methylated extensively with M6A, it doesn't do well. Why it doesn't do well is an interesting question. They're probably working on that. Maybe it doesn't translate. Maybe it's not. Maybe it's degraded, et cetera, et cetera. But now we know that there is a role for this in plant viruses, so that's neat.
Mm-hmm.
One of the things I found cool is at the very end of their discussion when, you can go off on kind of wild and crazy tangents. And this isn't that wild and crazy. It's just kind of fascinating. They talk about the fact that Gokhale et al. Have proposed that regulation of this M6A modification in HCV, hepatitis C virus, would allow the virus to replicate at low rates and maybe evade the host immune system, establishing persistent infections.
Right.
And then they talk about a plant family that actually has proteins with this domain encoded by the virus.
Yeah.
Sorry, a virus family that has this domain encoded by the virus. And these viruses infect
most of them infect woody plants.
Woody or perennial plants where they would have to establish infections that persist for years.
Yeah.
Mm-hmm.
Yeah.
And then they come back to this system for alfalfa mosaic virus. And they point out that, the alfalfa mosaic virus seems to have co-opted this host function, and alfalfa plants might last for as long as 5 years. So
yeah, it's cool.
I just thought that was a really cool tie-in to
remember
persistent virus.
The alfalfa capsid binds this demethylase. So maybe that's a way of recruiting it to the viral RNA to get rid of the methyls, because that's not good for viral replication in this case. Okay, cool. Now I'm sure Steen will write us. Mm-hmm. Steen often is.
We're hoping
he's a plant virologist. And
that's
he will
right.
correct us
for clarification.
And
that's
correct anything
exactly
that we said that's incorrect.
Yeah.
But I thought it was interesting. And at some point we'll do a mammalian virus paper about M6, right?
Okay.
But we never did one before, so here it is. Plants lead the way.
There you go.
They were here first.
Cool.
What do you expect?
What do you mean?
They were here before animals.
But not before bacteria.
No.
So first is not correct.
Bacteria have this too?
No, that's not what I meant.
Well, that's what I thought you meant.
Do they have a methylase?
Yeah, well,
that's a good question. Does anyone know?
Huh.
Adenine methylase bacteria. Let's see if Sir Google,
right?
Or Madam Google, or Thou Google. In bacteria, adenine methylation is part of the restriction modification system. There you go. Of course.
Oh, right. Methyl-sensitive restriction enzymes.
Yeah, exactly. There you go. Isn't it cool how you can find answers just by searching?
Yeah,
But what if you find an answer that's fake?
The
what do you
beauty
do?
of the internet is that everybody is only a fraction of a second away from a wrong answer.
That's
right.
True.
Or a right one.
That's right. Is that a referenced fact, by the way? Did you find out who said that?
All right. So this is great because, someone put in pictures of this alfalfa mosaic virus, and the capsid it looks like it polymerizes.
This is so bizarre.
And who put in the picture from the Japanese movie?
From Spirited Away.
Spirited Away.
When I saw this green illustration, I was immediately reminded of this this character from Spirited Away.
3 heads. Or
these 3 characters that bounce around as heads.
It's funny. Dickson, you probably don't see it because you're not refreshing your document.
I am not refreshing.
Now, Alan, what's the relevance of this virus on Massachusetts?
Oh, I drew that with an airplane.
You flew that, really?
Yes, yesterday morning I wanted to log some. I wanted to log
here it
some time.
Right.
So you flew an icosahedron.
Yeah, so I couldn't. I've done various maneuvers and such in this plane already, and I didn't really need to do any of those right away again. And I thought, what am I going to do? I don't really feel like flying someplace just for the heck of it. And then I suddenly was inspired, and so I put together a route and I flew a two-dimensional representation of an icosahedron superimposed over western Massachusetts.
It's nice. So you had to plan all this out ahead of time? Or you just
I did. I put a bunch of the GPS coordinates in. I used Google Earth to get what the coordinates should be. I superimposed a capsid drawing over Google Earth and then got the coordinates off it and put it into my EFB app. And then
is that the biggest virus known?
I like to think so. I like to think that I have synthesized a giant airborne virus.
My goodness.
It wouldn't be the biggest fungus known, right, Dickson?
No.
Those are thousands.
No,
of
right.
miles.
Those go on for miles.
They do.
And by the way, this is right next to the Quabbin Reservoir.
Oh, yeah.
So it's hopefully it won't drift into the water supply.
No. If you do, you'll certainly notice.
Nice.
Yes.
It'll displace half of the lake.
So this is from the program you're using, the app that you're using?
Yes, that's my flight track.
It's got emergency up there.
So this is the actual track that you flew, or what you plotted in advance?
That is the actual track that I flew. What I plotted in advance was more precise.
It looks good, doesn't it?
Great.
Next time you could do the genome inside.
Oh, people have been sending me suggestions for things to do since I posted this on Twitter. Actually, Column suggested that next time I should change altitude so that we can, get the 3D view.
Yeah, very good. Would it reflect in a thinner line by the altitude?
No. It's you'd have to do the whole thing.
So what altitude was this flown at?
3,000 feet.
Nice.
Which ends up, for some of the higher hills, that ends up only being about 1,500 feet above ground, but
cool.
It was stable enough air.
All right, let's do one round of email.
Mm-hmm.
The first one is from Erwin Dweezer.
Wasn't one of Frank Zappa's kids named?
Yeah, Dweezer.
Dweezel.
Dweezel.
This is different. This is a Dutch spelling.
Well, Erwin was a paper that one of the authors on a paper we did on TWiV 459. I listened to your podcast, TWiV 459. I want to thank you for the extra attention you paid to our paper on the wild poliovirus 2 incident. This was the spill in the Dutch plant. In discussing, you raised some points I can provide a bit more info on. Not too much, since we are preparing a paper on all the lab results, including quantification, and some of the production facility-related information is not for me to share. I am glad you noticed we did put in quite some effort to obscure the identity of the operator, which, if I remember, in the next episode was shattered by someone else who wrote it. Production facility. The aerosols were formed when a leak occurred in the tubing connected to a virus cultivation unit. Production facility is GAP3 compliant, including obligatory showering out, but no respiratory masks were worn since the production process is overall a completely closed process. Cool. Vaccination. Please realize that also with Sabin vaccine, you can get infected and shed polio. Sure, the probability of infection after exposure is a bit lower, and shedding is definitely shorter and of lower titer, but really there's no guarantee to not become infected after Sabin vaccination. In the Netherlands, we stopped providing OPV to polio vaccine production facility employees about 3 years ago. Sewage. We realized from the start that shedding in the sewage was not okay with respect to GAP3, but we did not have anything else up and running to contain stools at that moment that would improve the situation for both GAP3 and public health. Realized that the removal of polio 2 from the OPV had no effect at all in the Netherlands since we never have used OPV, so we have no decline in protection against polio 2. On the long-term detection in the sewage system, we sample really small populations in the sewage, a couple of hundred households, with the detection limits as in our paper on environmental surveillance, which she provides a reference for, and some modeling as in risk assessment, risk management, and risk-based monitoring following a reported accidental release of polio in Belgium. That's another paper. We estimated that 3 to 6 weeks detectability was likely. We briefly considered polio excretion in urine, but since we did not find any hint pointing at this being relevant, we dropped it from our to-do list. I am very interested if you have info that this might occur.
Isn't that a do-do list?
It's urine, dude. It's urine.
Sorry.
I don't have any info that might occur. We were just curious, I think. I send you this email from a partly foggy Bilthoven, but forecast promises temperatures of 20 C, slightly above normal, and with almost no wind. It is quite pleasant to go ride my rowing bike. Check rowingbike.com.
Rowing bike.
Rowing bike. I know. I have to know what that is. Well, it's loading.
It's like a recovery bike.
Yeah, but you use your arms.
Yeah, I've seen those.
Yeah,
they're cool. Very
I've seen people on the road with those.
So I have a personal message for Erwin if he could say hello to a good friend of mine who works there also. Her name is Joko van de Giesen. I'm sure you know her. I'm sure you know her.
Is this in the Beethoven?
Yeah. Mm-hmm.
So Erwin is head of the section of enteric viruses in the Center for Infectious Diseases Control, the National Institute for Public Health and Environment in Beethoven.
Right.
Thank you, Erwin. We appreciate it.
It's a great place.
Dickson, can you take the next one?
Sure. I can barely read it. It says, Yonne writes, from last year, great special in Delhi. And then I can't read this. It's got some chikungunya.
So this is
there's a meal here.
Chikungunya. See it?
I see malaria there too, though.
So these are prices for diagnostic tests.
Got it.
So Johnny is our pediatrician friend from Cambridge.
So you can get tested for malaria,
that's
for
right.
a certain number of rupees, and for chikungunya.
So the deal, rupees 500.
Boy,
the comprehensive wellness is 3,000 rupees. Special price, though. They're all special prices.
But for you, that's right.
And 500 rupees are $7.73 US. Very reasonable for an order of chikungunya.
Exactly. That's with or without rice.
Add some rice on the side and some.
Of course, hot sauce.
What's the cheapest for the diagnostic test you could find here in the U.S.? Say malaria diagnostic. How much would that cost?
Are you kidding?
Hundreds, right?
Absolutely.
And it's 500 rupees.
Well, we don't have much malaria here, but they sure do where he is. Where this came from, they have a lot
nothing is $7 in this country.
Nothing is $7. That's true. Oh, wait a minute. If you go to the airport, you can order a Coca-Cola for that much.
Right. And not a diagnostic test.
I know.
Alan, can you take the next one?
Sure. John writes, Dear TWiViridae, I hope all is well in the TWiFth dimension.
I like that.
That's not bad.
That's good. Why is it that particle to PFU ratio is in common usage and not efficiency of plating? Personally, I find it easier to think of efficiency of viral infectivity as 1 PFU in X number of particles. Given particle to PFU is in common usage, I think I might be missing something. It's 14 degrees C and raining in Melbourne, Australia. Thank you for all you do. May the infectivity of your podcasts be ever as efficient as Semliki forest virus. PFU equals constitutive listeners.
Nice. How efficient is the Semliki forest virus? 1 to 1. Wow.
He must have listened to some virology lectures to know that.
Yes.
Goodness.
Well, I don't know why. I asked Lynn Enquist, efficiency of plating, as John says, is the virus titer in PFU per mil divided by the number of particles in the sample. And the particle to PFU is just the inverse. So I think EOP was historically first. The phage people first started using that, and then it changed. And I don't know why. Lynn Enquist didn't know why. Do you know why, Kathy? Why?
No.
No idea. So you're not missing anything, John. It's one of those things, just the way it is.
That's right. It's human nature or something like that.
Although maybe someone knows. Maybe there's someone out there that's older than Dickson that might know.
No, nobody's older than I am.
Maybe somebody was in the smoke-filled room when they killed efficiency of plating.
That's right, when viruses were first described. So is Semliki forest virus the most efficient of all the viruses?
Well, you can't get better than one, can you?
No, but are other viruses also as efficient?
There's probably others out there. But on our table in the book, that's the best one.
That's the best one. Isn't that interesting? And is there a particular reason?
In fact, Alan found it by Googling a couple of weeks ago.
But do we know why it's so efficient? So they produce very few defective particles when they replicate, right?
Don't know why. But that's why Ari Helenius, way back when he started studying virus entry using Semliki virus, he was using microscopy, so he could say that particle is infectious.
I see what you mean.
As opposed to a virus where 1 in 10,000, right? Who's next? I forgot. Alan, did you do one?
I just went.
Kathy, can you do the next, please?
Sammer writes, Hello, thanks for doing these podcasts. I've enjoyed them quite a bit. I have 2 points about animal-related comments made during TWiV 458. First, regarding the exudate from the cut surface of the growths in rabbits infected with the myxomatosis leporipox virus. This is not pus, purulent suppurative exudate, but in fact a proteoglycan matrix produced by the myxoma cells, undifferentiated mesenchymal cells, and does very much resemble mucus and is often referred to as mucinous in pathological descriptions, and he gives a link. Second, Dickson said that leopards do not have molars. Felids do, in fact, have small molars, but usually only one, and they are not used for chewing in the same way as herbivores and omnivores.
Right.
And he gives a link about that.
Right.
Best Simmer.
Can I be pedantic?
Yeah, sure.
I'd love to, because you pronounced, where were we again? Mesenchymal isn't mesenchymal. It's mesenchymal.
It's not mesenchymal?
No.
I like mesenchymal.
But it's mesenchymal. I'm sorry.
Is it falciparum or falciparum?
It's both, because the British rule, but they don't say mesenchymal. They don't say mesenchymal. They say mesenchymal.
Sammer is a doctor of veterinary medicine, so I think this person is right.
I'm going to send him the picture of my leopard with the mouth wide open, and he can tell for himself.
No, this person.
I would love to see. Yeah, no, I'm going to send it to him.
No, Sommer.
To this person, to this writer.
Exactly.
Not
who's on first.
We don't know if it's him or her, is what they're saying.
Oh, I see what your point is.
Dickson, send me the picture. I'll send it along to Sommer.
I'll do it.
Okay. You want to follow with Rich's comments, Kathy?
Oh, Rich said that he had written to Grant, being Grant McFadden, about Mark's question about the name Myxoma before TWiV 459, where we read Mark's follow-up letter. Grant's response came in too late for the show. Sommer's letter provides a good segue to Grant's response, which is, the virus lesions were originally described as looking like mucinous tumors, resembling various cancers, but it really wasn't mucus the way we understand it now, and it certainly isn't cancer. However, the bacterial infections in the eyes and nares does cause the secretion of runny pus-like gunk and extensive conjunctivitis. I think myxoma name came from the mixed tumor-like appearance of the primary lesions.
Well, that's quite an arc that we've had with that mucus.
Truly.
An arc of mucus.
An arc of mucus, that's right.
Let's do some picks. Mm-hmm. Alan, what do you have?
I have an article that, since we did a plant virus this episode, I thought this would be a good time to pick this. It's about farming in the Netherlands, and I'm sure Dickson will enjoy this. It's a nicely done article in National Geographic about how the Netherlands has, over the past couple of decades, it decided to convert all of their farming to super high efficiency, mostly indoor production.
That's right.
And as a result, they have become the leading European source of many fruits and vegetables, including things like tomatoes. So this has become a major cash industry for the Netherlands, that they've got these high efficiency farms, and because they're right there in Europe, they have shorter transit routes than you would have by importing a lot of these things from more tropical places. And it's a well illustrated article as well.
Yeah, I've seen it.
Gorgeous photos of these just acres of greenhouses extending across the flat lands that they have there.
Alan, what's the solution to their problem when the demand exceeds the production?
Well, their solution is to go up.
Yeah, that's right. Exactly right.
They go vertical.
Yeah, they do. Yeah, they do.
Vertical farming going on that they're feeding people with. The title of the piece is a little misleading. This tiny country feeds the world.
Yeah,
they don't actually feed the world. And of course, they've emphasized the high profit margin crops like tomatoes. I'll be a lot more impressed when somebody figures out a high efficiency way to grow beans and rice. But
true.
This is certainly, a move in that direction, and they're doing some really cool stuff and some cool crop science that they are openly sharing with a lot of developing countries where this is sorely needed.
Sure.
And I thought it was just a really cool.
That's fabulous. That you
I love this photo of the farmer's house next to all the green.
Yeah.
Which is probably done by a drone, I would guess.
Yeah, probably.
Yes. Yeah, there's a lot of drone photography in this.
So can I follow up on this just for a moment? Because I don't mean to pat myself on the back or anything like this. Please don't think that I'm doing it. But I just happened to have been at the United Nations yesterday to participate in a conference on, it was called The Future of Everything, but to be honest, it was all about artificial intelligence. And they included me in there because there's a lot of automation involved in some of the vertical farms in, let's say, Japan and certain places like that. But the guy who arranged the meeting was a bureaucrat at the United Nations from the Netherlands. And his name was Job. And Job and I bonded deeply. And he told me about this particular thing. And he said, I can't believe that we haven't gone higher yet, because the Dutch can sell all they grow. And he says this conference will alert them to the fact that this is going to happen. Okay, so we should be first, because look at what they've done already.
They have some cool infographics here. One showing the square miles of greenhouses in the Netherlands. 36 exceeds the square miles of Manhattan, which is 23.
Yes, exactly.
It's pretty cool.
Yeah, it's very cool.
Joop.
Joop, J-O-O-P.
I like Joop.
Joop.
Dickson, do you have a pick?
I do. I ran across this in, of course, Cosmos magazine, because I now subscribe. And I read the headlines, and then I go to the articles sometimes, but not mostly.
Can I buy you a few other subscriptions?
Absolutely.
So that you
No, come on.
Beware the man
I'm
who
not shilling
subscribes to
for
only
this
one
magazine. All right, go ahead. Heap it on. Go ahead. Just guilty as charged. But here's an article that's absolutely riveting, that suggests strongly that almost all of the plastics that ends up in the oceans only comes from just 10 rivers.
Really?
10 rivers, and they're all in Asia.
I thought we dumped plastic from boats.
Yeah, you thought so. That's nothing compared to what they do.
They just dump them in the river?
Yeah.
Why don't they burn them or something?
If you've traveled throughout Asia and South Asia and Southeast Asia, you can see it for yourself. It's just overwhelming. And as soon as the rains come and the monsoons come, all that gets swept out to sea. And then it ends up in these gyres.
Gyres?
Gyres.
They exist?
They do, of course. And no one has found a way to get rid of them yet. And they suggest strongly that because plastic has a certain sweet flavor to it, apparently, to birds, birds are particularly susceptible to the ill effects of these little particles that end up being produced by plastics just rubbing up against each other.
Well, and fish and sea turtles.
Yeah. So I was very disheartened when I saw the article, but I thought maybe by seeing all of this for the first time.
I saw this news as well. I didn't see it in Cosmos magazine, but it was widely cited when the research came out. And I think this actually is kind of a positive thing, because it means if this is correct, if most of this plastic is coming from these 10 rivers and we know where they are, we could intervene. And you could do things like try and catch it before it gets out the mouths of these rivers, which at which point it's in a much smaller concentration and you could round it up more easily. So it could be a hopeful story.
I just refreshed Dickson's iPad so he could see the pronunciation.
Yes.
And it's mesenchymal.
That's the third pronunciation.
I have never heard it any other way.
The first one is mesenchymal.
Mesenchymal.
Well, so much for pedantticism.
So what are you going to do, Dickson?
I'm going to just say, okay, that's the way it is.
Back on the river pollution thing, on social media this week, I found out something else that we should all feel horrified about, evidently. That is microfiber pollution. And every time you wash any kind of synthetic clothes in your washing machine, you're putting all these microfibers in the environment.
Lord.
And, we're already eating them, and they're everywhere. And So. But there are devices evidently you can throw in your washing machine to capture them.
Really?
Whether they're at all, whether they're snake oil. Yeah, I don't know.
Dear.
Kathy, what do you have for us?
I picked a book that's an ebook from NASA. It's probably a regular book too, by an author named Michael Meltzer, who I think I misspelled his name here in our Google Doc, but he was a planetary protection officer at NASA. And the reason this came about is that the Cell and Molecular Biology retreat last week for their plenary speaker had Catherine Conley, who was also a NASA planetary protection officer. And so this book is When Biospheres Collide, and it's a book describing the importance of being aware of how difficult it will be to detect life elsewhere as soon as we get there, because we will contaminate wherever we landed. And I had, now I can't find it. Oh, here's my version that I marked up, I think, that I wanted to read. And yes, Planetary protection addresses the real consequences that might result from an actual transfer of biological material between different planets, if indeed life does exist on other worlds. And then, this is the cool thing, this particular author said, being the NASA Planetary Protection Officer, it's my job to ensure that our space exploration efforts neither unintentionally contaminate other solar system bodies nor Earth upon spacecraft's return. However, my interest in this is much more personal. So his research on muscle atrophy was something that was had an experiment that was carried on the Space Shuttle Columbia that tragically disintegrated during re-entry on February 1st, 2003. But when they reopened the recovered hardware several months after the accident, many of the experimental animals had survived the crash, demonstrating that, if properly shielded, even multicellular organisms could survive a meteoritic style entry event.
Okay, stop it already.
Required for the natural transport of living organisms. So anyway, it's kind of cool. And when I talked to her, or either when I talked to her afterward, or Catherine Conley, or in her talk, she mentioned that I think it was C. elegans was an experiment that she had, or maybe that's what she's referring to here. Anyway, that some experiment survived. So anyway, it's a real concern, and evidently there's now less concern at NASA than there used to be about this kind of issue. And in fact, the Planetary Protection Committee that my colleague Mike Imperiali is on hasn't met since 2016, she said. So I don't know.
This would be interesting to see a follow-up to the movie The Martian.
Yeah.
To see what happened to all those potatoes.
Yeah, this is a whole long book, and it's available as a free PDF at NASA. So if you're interested in more of it, check it out.
Cool. That's pretty neat.
Right.
If you need to know how to pronounce something, ask Dickson.
Right, exactly. No, ask Dickson.
I used to call you Dick, remember?
You did.
For the longest time.
I used to call you Vince.
I'm not.
Then you said, I'm not Vince, I'm Vincent. And then I said, in that case, I'm not Dick, I'm Dickson. So that's where the chemistry started.
Well, you were happy to be called Dick for a long time.
Most of my friends do.
And then all of a sudden you said, I don't like Dick.
I got the biggest laugh of any particular person in Australia, I'm sure, when they introduced me on this television show that I was dragged off to. They said, what's your name and where are you from? And he says, from the U.S. He says, what's your name? And I said, Dick. The entire audience absolutely went hysterical.
Of course.
It was like a gumboots joke. I mean, come on, people, grow up.
Oh, are you telling the Australians?
Grow up? No, you can't do that because they won't listen.
I have a pick, which is a podcast from the American Society for Microbiology. It's a revival of a podcast originally started by Mary Buckley called Meet the Scientist. She did a very nice job for many years. Then she left, and Carl Zimmer did it for a while. Right. And it stopped a number of years ago, and now it's been revived as Meet the Microbiologist. Ah. And the interviewer is Julie Wolfe, who has been the ASM Communications social media specialist for a number of years. And she does the interviewing. And so it's pretty neat. She does, I think, 2 individuals per episode, or maybe just one, I don't remember. But anyway, the first one is MTM 65. So that's how there were 64 previous episodes. She's starting at 65 with a brand new name. And, she interviewed me. She came up here because she's here in New York City. I gave her some advice at recording and so forth. And she talked to me. So you have all probably had enough of me, but there are other people in subsequent episodes that are going to be interesting. So I would check it out. It's good. It's produced by Ray Ortega,
cool,
who is our friend at ASM. So check that out. Meet the Microbiologist, asm.org/MTM. Nice URL. We have a couple of listener picks. We had one from way at the top of the show from, who was it, Jay? He picked Omega Tau Science and Engineering Podcast at omegatauPodcast.net. Some in German, some in English. And so that's a podcast I've known. Dickson and I were interviewed on it many years ago. Remember, Dickson?
I do.
He said, I want to interview you and Dickson about viruses, bacteria, and parasites in a half hour.
Exactly right.
That was so funny.
And with time for questions afterwards, too.
It was funny. All right, so that's one pick. And then we have another one. here from, Trudy. Dear TWiVers, upon Vincent's recommendation, I read The Beak of the Finch and Time Love Memory by Jonathan Weiner. What I love about reading good books is that they often lead to other good books. Time Love Memory repeatedly mentions Aerosmith, a 1925 novel by Sinclair Lewis about the culture of science. Only about a third of the way through, but I'm really enjoying it.
Yeah.
So far, I would like to suggest it as a pick. Sure. I picked it on TWiV 11.
I was going to say, I remember that being picked a long time ago.
And I also included a page. I had my mother's copy. She was an English teacher and taught this every year, and I had one of her marked up pages with brackets and asterisks.
Cool.
I still have her copy of the book. She loved it. She absolutely loved it, and it's a great book, of course. So, yes, that's good. And one more from Mark. Hello to the TWiV Quadrangle. Generic follow-up. You are all doing a great job. Keep up the good work. 2 listener picks that your audience might enjoy. One is an iOS app called Human Anatomy Atlas. The app runs on iPhone or iPad, provides a 3D model of the human body.
Wow.
Users can explore specific systems, skeleton, lymphatic, digestive, etc. You can do a virtual dissection. You can display notes. 99 cents.
Wow.
2, Master in Business podcast, produced by Bloomberg, by its host Barry Ritholtz in Midtown Manhattan. Each episode's an interview with someone having a distinguished career in finance, which is kind of an oxymoron, in my view. So they're all crooked.
Some have to Skype in from
That's
prison.
where the money is.
They have to Skype in from prison.
They're all alive because of science. Guests range from Nobel laureates like William Sharp, Robert Shiller, Daniel Kahneman, investment innovators, and others. In a style analogous to TWiV, the host asks the guests about their backgrounds, how their career started, and the work they do in finance. He has a few stock questions that get lots of insight. What do you know today that you wish you knew when you were starting your career? I have to use that one. And what would you tell a millennial or new graduate wanting to enter your field? All the best, Mark. P.S. The weather this week in the Bay Area has been glorious. Temperatures in the mid-70s, clear skies and warm sun. P.P.S. Continue being nice to Dickson. P.P.P.S. Let me be mean to Dickson by asking if urban agriculture will ever have a new show.
The answer is yes, it will. Yes, it will. You bet it will.
30 years.
Come on, we're going to do one soon.
Soon?
Soon, very soon.
All right, that's TWiV. You can find it at Apple Podcasts, microbe.tv/TWiV. Questions and comments, TWiV at microbe.tv. Financial support is welcome. Go to microbe.tv/contribute. Dickson Despommier can be found at thelivingriver.org, where you have put up new pictures. Is that right?
No, that was on my photo website.
DespommierPhotoArt.com.
Yep.
Wow, you have a media empire.
No, it's your fault. You're in facilitating me for these things. Come on. You
parasiticdiseases.org, where you can get a free copy of 6th edition of Parasitic Disease.
Parasites Without Borders.
Parasites Without, what did I say?
You said Parasitic Diseases.
That works too. I made that one.
That'll work.
And you have a free PDF in English and Spanish, right?
Correct.
Amazing.
Yep.
You're giving them away.
We are.
Wow.
That's what
great.
we should be doing.
Thank you, Dickson.
You're welcome.
Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Alan Dove. AlanDove.com. On Twitter, he's Alan Dove. Turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiV. I'd also like to thank the sponsors of TWiV, Blue Apron and the Defense Threat Reduction Agency. The introductory music on TWiV is composed and performed by Ronald Jenkins. You can find more of his work at ronaldjenkins.com. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.